Well, thanks so much, Ian, for the kind introduction and also the JGI for the opportunity to be here. It's been a really inspiring 3 days and so many examples of cool biology. I'm always very fascinated and interested in the scales biology can work with, especially looking at from an application side of view, and also that it can be programmable. As society, of course, I think we make use of biology for biomanufacturing since thousands of years. And also at the turn of the last centuries, and I think we discovered that we can actually also make chemicals using biology. I think pioneering works there from Chaim Weizmann, who discovered this acetone butanol ethanol fermentation, which at its heydays was used at massive scale, and also became very decisive during the world wars, as acetone was used for making cordite for gunpowder. And actually, Chaim Weizmann later on became the first president of Israel in recognition for his work. Fast forward 100 years, I think the ABE fermentation became phased out, but I think in general, I think we became really good at biomanufacturing, and especially I think biology can operate in a distributed way compared to, I think, the chemical industry. And building on that, I would argue that I think biology is actually uniquely suited for some of the biggest challenges we face as a society today, with unprecedented levels of CO2. And then also, I think, the accumulation of plastics and other waste streams, for which I think we have a very, very small marginal recycling loop right now. But if you look at those industrial waste emissions or greenhouse gas emissions, and also I think if you gasify some of those wastes, it's actually not so different from hydrothermal vents that you would find at deep sea. And that's actually very speculated that life on Earth started. And you can see here, this is the Wood-Lungdal pathway, which I think is believed to be one of the oldest pathways on Earth, which is able to fix two molecules of either CO or CO2 into acetyl-CoA, one of the building blocks of life. And this pathway is still believed to be the most efficient CO2 fixation pathway to date. At LanzaTech, we use an organism that has this Wood-Lungdal pathway, an anaerobic acetogen called Clostridium autoheterogenome, which was isolated from rabbit droppings and is able to produce ethanol from carbon monoxide or carbon dioxide and hydrogen. At LanzaTech, we've built a gas fermentation process around this organism, and the process is able to use a variety of feedstocks as input. This can be industrial off-gases, for example, flue gases from the steel industry or ferroalloy industry or the heavy industry. It can also be any kind of solid waste that can be gasified. So think about agriculture waste or also urban municipal waste, or actually CO2 directly with, for example, renewable electricity or green hydrogen, and can, for example, also be integrated as direct air capture. At the back end, I think once the carbon is fixed, I think we can produce and turn that into ethanol, which I think can be used as fuel. The biomass itself is a rich source of protein, but we can also use and make a variety of chemicals and materials, and then at the end of the life, we can bring those back and close that circle. So we spent over 15 years to scale this process from bench scale all the way to full commercial scale with tanks of over 500,000 liters in size, and operating now as several commercial plants. Our first commercial plant we started in 2018, and we already produced over 40 million gallons of ethanol. And by doing that, we're able to avoid over 200,000 tons of CO2 emissions going into the atmosphere. We also just started a municipal solid waste ethanol plant in Japan, and I think this video here gives you also a sense of the scale of those plants. So one of the key benefits that biology offers in this is, I think, that it can really use chaotic input streams. I think if you look at catalysis, there's the Fischer-Tropsch process, which also can kind of convert syn gases, for example, but it can only use very steady gases. So biology is really tolerant. It can use chaotic input streams. It can tolerate some contaminants. So this data here is data from, I think, continuous fermentation from, I think, June 2020 to February 2021, so over 8 months. You can see, I think, the CO contents can vary quite significantly in that gas we get from the steel mill. But the ethanol production is always very steady. If you go to something like municipal solid waste, so here an example from our plant in Japan, I think depending on what's in the trash on a given day, I think you can imagine that that gas fluctuates even more. But again, we can generate ethanol very high selectivity, and they're very stable. So we're now in the process of, in addition to the plants we already operating, I think starting over the next year, 6 more plants, and there's several other plants now already in the late stages of engineering. So I think very soon we'll have the ability to capture over 1,000 tons of, over 1 million tons of CO2 and produce over 600,000 tons of products, and do that on a global scale, but always with regional inputs. I think those inputs can be the heavy industry, various waste feedstocks, and also CO2. So I think you find those pretty much anywhere on the globe. And those industries are at the moment responsible for 12 gigatons of carbon emissions, and have having very mandates to reduce their emissions. At the same time, with the products we can make, I think those are in high demand, especially having sustainable solutions. So I think we kind of can combine 2 very different markets here and create quite new value chains in that sense. On the product side, ethanol is our first product, which can be used as fuel or also in cleaners or fragrances directly. But we can also develop technology with partners to turn ethanol to sustainable aviation fuel. We actually spun that out into a separate company called LanzaTech, and also can feed into the ethylene value chain with polyethylene, PET to make fibers, materials, and also surfactants, which we call carbon smart products. Now, synthetic biology also offers us to modify the organism and make other products directly in our fermentation. It can be acetone, can be propanol, can be monoethylene glycol. In fact, I think we've already shown over 100 different molecules that we can make with the organism directly. The key challenge at the beginning was when we started, there weren't any genetic tools available for this organism. In fact, only in 2010, I think the first publication of a successful genetic modification of an acetogen was published in the first genome sequence. And also, I think the organisms, the biochemistry was understood, but not so much the molecular mechanisms, and also parts of the metabolism were still a black box. I think we had to together with partners, I think, develop an understanding of the metabolism, and also then develop more and more advanced tools, and also some genome scale models and other models that we can predict the metabolism. So unfortunately, I don't have time to go into all of these in detail, but I want to show you at least some of the ways we apply those tools then to bring a process from a proof of concept level to an industrial scale level, so from milligrams of production to grams per liter per hour production, and really high selectivities. And if we think about that, I think we think about that in, on one hand, on how can we optimize a pathway, identify the best enzyme combinations, and also how do we optimize the strain to channel flux into the pathway of interest and also avoid leakages from there. And then, of course, how do we scale that up and transition that into the field and develop this on a continuous production level. So I think I started with Chaim Weitzman. When we started out the company, I think we had the luck that I think David Jones, a professor in New Zealand, and the company started out in New Zealand, had this collection of several hundred Clostridium strains that span several decades of industrial development before the process was stopped. I think you can see some of those violets all the way back to the 1940s. And you can also see they come in various forms and shapes, those those collections. So initially we hope, can any of those strains maybe grow on those guest streams we're interested in, since it's also Clostridia strains. Unfortunately, it wasn't the case. So this box was sitting in the corners there for 10 years or more. But then was the advent of sequencing technology and the opportunity to partner with the JGI. We then went ahead and sequenced this whole collection. So one challenge, of course, was getting that collection to grow and high quality of DNA. So we had help from Oak Ridge with that as well. But we're actually able to grow 90% of those strains and generate DNA that we could use for packed biosequencing. So in the end, I think we produced, over 270 genomes, I think mostly Clostridium biorhynchia, but also some other species. And then mined those genomes for genes of interest, and those genomes are public now. So I think we've identified several new protein families. In particular, we were interested in the biosynthesis genes for making acetone or butanol. So I think we're able to identify a few unique genes there as well. My colleague Nick actually also did some DAP-seq work together with the JGI to identify some transcription factor binding sites. I think if you're interested, I think Nick is still here, and I think the poster might still be online at least. But coming back to the acetone pathways, I think we've identified over 50 unique biosynthesis genes there. You can see most of them were maybe just point mutations. There were some that are quite distant. So going in, we didn't know, would any of those be better than the wild type genes, and which one would produce. So I think we just combinatorial take randomly different combinations of those, also use different promoter strengths to express these, and then generated a combinatorial library that we transformed into the organism. And we screened about 250 strains and could see significant improvement over our starting point. Now 250 strains is already a lot if you have to do that manually and then as a scientist. So I think now we've also developed a system where we can automatically generate strains and screen strains. In fact, we can now screen thousands of strains in this system that you can see here. The key challenge here was that we had to do this anaerobic. So I think this whole system sits in an anaerobic environment. And also we want to do the screening in context of those gases like carbon monoxide and hydrogen, which are toxic and flammable. So I think we had to develop some solutions there to enable that. In parallel to developing the system that allows us to screen thousands of strains, I think we've also partnered with Mike Jewett's lab at Northwestern. So Mike has developed some technology to make proteins in a cell-free system, so using cell-free protein synthesis. So all you have to do is have your DNA on the plasmid and you can make your protein in vitro. So we thought this might be an opportunity to prototype pathways quicker. So I think for butanol, we applied this approach. So essentially what we made each enzyme variant and then did the same mix and match those. But we could do that more quickly, I think, just with liquid handling, essentially. So we started with one combination of genes and then just substituted one gene at a time and used different levels there of expression. You can see some of those genes don't produce at all, some only produce at higher levels. And based on this base set, then we could design the next set where we chose the best enzymes and we could actually find an improvement. But we've also applied some machine learning techniques and those actually outperformed on the data sets, even kind of our expert scientists from actually both our labs and Mike's lab. So we could find a pathway very rapidly that was 4 times better than this base case as well. So the key question then remained, how does that translate to in vivo? So is that really predictive? And as you can see here, we've done that for now 3 different pathways, and it's quite a good correlation. So I think in particular, what you can see, everything that doesn't work well in vitro also doesn't work well in vivo. So I think you can already reduce it to large design space. And we've published this work in Nature Chemical Biology and more recently a follow-up paper in Nature Communications. But the beauty about this approach is that you can do the screening here within a couple of days instead of having to grow the cells, which takes weeks. But one challenge we had, and if we go from these results and into the cell, we had to again codon optimize synthesis, develop those in vitro constructs. So we thought, can we optimize this and do this quicker? So, and together with the JGIs, and we've developed a modified vector system where we took the cell-free vectors that Mike's lab used and introduced the golden gate sites. And those golden gate sites, I think we tried different combinations and found some that didn't affect the cell-free testing. We then also developed a set of donor vectors that had different genetic parts, like promoters of different strengths, and we could pull in some data we generated with Howard Zellis' lab, where we validated over 1,000 promoters, barcoded promoter sequences. So combining those best combinations from the cell-free was various parts, and then a expression system for our host. And we've developed an automated system where we can do that very quickly. We can now go very quickly from those cell-free vectors directly into in vivo assemblies. And those vectors are available through Genome as well. But having an optimized pathway alone is not everything. So I think when we put this optimized acetone pathway, for example, in the cell, what we saw very little acetone, but still a lot of ethanol and a lot of isopropanol actually, and some 3-hydroxybutyrate. So isopropanol and 3-hydroxybutyrate are not normally produced in the cells. I think we thought this comes from interaction of the native metabolism with the pathway we introduced. And computationally, we could predict there's a couple dehydrogenases, reductases, tauesterases, which potentially play a role here. But even the short list had 15 enzymes. I think knocking those out in combinatorial ways is still something that I think takes a lot of time. So again, we thought, could we look at our cell-free system? So I think we've made a cell-free system that can make acetone and then spike in one of those effector enzymes at a time. So most didn't have an effect on acetone production, but we've identified a couple that where we saw some 3-HBs and or isopropanol produced. We did some omics analysis as well. This was Oak Ridge, where we could see that one of those identified genes is actually not really expressed. So I think we focused on the others one first. And knocking out these 3, we had identified as a short list. We could then very quickly optimize acetone production to about 90% selectivity. To really optimize this further and kind of go through this quicker again, we're now in the process with the JGI to build out a single gene knockout library for this Clostridium organism we using, where the JGI is assembling the knockout constructs. We're going to generate these knockout strains, and we have a partner in Australia, the Synthetic Biology Center of Excellence there, that will do multi-omics analysis on all of these strains. I think once you have a target identified, we already have a collection of strains available. And we think this collection will also be really useful to characterize the metabolism, for example. We've also developed a technology to do CRISPR-I, so I think essentially reducing expression of genes. So I think we published the proof of concept for that, and now together with the JGI developed a CRISPR-I library. So I think this library has about 12,000 guide RNAs. I think about 3 per genes that we selected. And you can already see here when we transform this into the cells, we already see a strong biological influence. But the way we can use these libraries now is, I think, do fitness experiments. I think, for example, we can challenge the cells with acetone stress. And then I think at the start, I think we have a kind of distribution of the libraries. And then at the end, we can see which of the guides are enriched, and then kind of go into those targets and I think modify those targets in our strains. So I think this is a very useful tool to identify targets on a genome scale level. So having then a strain or strains that can produce acetone or isopropanol very well, I think we've developed a continuous fermentation process, and we're able to show rates of over 3 grams per liter per hour, 90% selectivity with several weeks of continuous production, actually that already in an in-field pilot plant we have, and I think looking at rolling that out commercially next. What we also did, and I think carbon is very important for us, I think we did some life cycle analysis with that. So I think acetone right now is exclusively produced from fossil sources, and you can see per kilogram of acetone you produce, you release over 2.5 kilograms of CO2 into the atmosphere during the manufacturing process. With our process, we cannot not even be carbon neutral, but we can even be carbon negative. So I think we can lock in CO2 from the atmosphere into the acetone product. So almost 1.8 kilogram of acetone of CO2 can be locked into per kilogram of acetone produced. So if you make then, for example, acrylic glass from acetone, I think that carbon is there locked in for a long time. And we published this work recently in Nature Biotechnology as well. So we've already produced over 100 different molecules. So I think these span not only kind of those shorter chain molecules, but also longer chain molecules. And we're in the process now optimizing some of those. And really the idea is to have strains at hand that can produce different products, and then we can then kind of react on market needs by just changing out the microbe. And the advantage, of course, we have is our process using a waste that's not treated as commodities. So I think we're kind of off those cycles. We've already have now, which is I think very exciting for us, the first products in consumer hands made from those waste resources. That includes various kind of detergents and cleaners, for example, with Unilever, fragrances with Coty, Job, Nivea. Also, I think packaging with L'Oreal and Mibel, and also I think materials and fibers and foams. For example, we have a dress with Sarah, collection with Lululemon. Shoes with On Running are coming soon, and I think also H&M. And then also I think we have the jet fuel where we already had transatlantic, transpacific commercial flights. Really, I think the key motivation for us is enabling a circular economy where we can capture and use those carbon or those waste sources, replace fuels on our everyday products, and then at the end of the life, bring those back and close that cycle and produce virgin products again. We're also hiring, so if you know anyone that's interested, then please spread that word. You're not looking for a job, but generally interested in climate or sustainability. I think together with the Engineering Biology Research Consortium, EBRC, we're also working on a technical roadmap around what synthetic biology can do around climate sustainability. Both, I think, looking at ways to sequester greenhouse gases, but also mitigating kind of climate change. So if you're interested, I think this is in the very late stages, but I think we're still looking for input and feedback. So I think feel free to talk to me or reach out to the EBRC. And with that, I'll thank you for your attention.
Excellent. Thank you very much, Michael. Any questions?
So obviously people have been working on this problem for a long time. Why do you think LanzaTech has succeeded where others have not?
it's a good question. I mean, obviously, I think it's the company is now started in 2005, so I think we're kind of 17-year journey already, but and now at a stage very at commercial scale. I think certainly I think scaling up I think is I think challenging, especially if you have to do that in kind of the field and with new feedstocks. So I think that's certainly been challenging. I think one thing that I think we did recognize very early, we need to be in the field. So I think already 2008 we started going to build a pilot plant when I think the process in the lab was still very early stage, and I think also operated directly on those kind of real world feedstocks. And I think we also obviously started out with a already existing product in ethanol. I think we're now adding additional products. I think that's certainly lessons that I think helped us. But yeah, it's obviously challenging, and especially I think getting through this kind of valley of death in terms of being able to commercialize the technology. I think once you're through that, I think it's maybe becomes easier as I think there's more adapters. But yeah, it certainly, yeah, we also partnered with a lot of different groups, which I think maybe also helped. So not just one pass. So these maybe things that I could share.
Okay. Any other questions? You're making me run, Nigel.
What I maybe also say in regard of the previous question, I think also on developing this strain and the tools for this strain, I think we partnered with a lot of academic groups and also JGIs. I think those partnerships were, and also the funding we got for those, I think were really important in a way.
So Michael, you showed data here, and you're able to run the process continuously for a very long time. Steps that you have to take be able to do this over 180 days.
Yeah, no, I think gas fermentation lends itself quite well for a continuous process. I think we have to sparge those gases into the reactor all the time anyways. And then I think also using those CO2 as a feedstocks, there's not too many organisms which compete with you. I think we have an advantage there that there's no, compared to growth and sugars where every organism can grow, that I think we have less contamination. I think ethanol is as an further advantage that it can be coupled to growth as well. But yeah, I think it's a lot of also how we run the fermentation regime. But I think if you can run a continuous process, I think it obviously has a lot of advantages in terms of uptime, and for gas fermentation, it lends itself really well.
All right. With that, thank you again, Michael. And I'd like to close the session and thank all speakers.